Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is going to cover the Electroexam 3 group problems. So let's start with number 1. Because the heart is autorhythmic, so break that word down, autorhythmic, it's got its own rhythm, the wave of electricity causing the beat arises spontaneously from the SA node. This is a bonus question, how does that wave of electricity move from one cardiac muscle cell to the next cardiac muscle cell? There's direct cytoplasmic connections via gap junctions in something called the intercalated discs, which are found between different cardiac muscle cells. What's the advantage of this? Well, it allows for a wave. It allows for coordinated contraction of all the muscles. However, there are many intrinsic and extrinsic factors that exert control over the heart. Specifically discuss the important intrinsic and extrinsic factors that can alter the chronotropic state of the heart. So chronotropic, what does that mean? That's speed. That's the heart rate. Chronotropic state is the heart rate. So this question specifically deals with the heart and how it can change rate through various influences. In terms of mean arterial blood control, the rate, stroke volume, and total peripheral resistance are important. So we're focusing on the intrinsic and extrinsic controls for heart rate. Pause right now. What are the intrinsic controls for heart rate for the heart? What are the extrinsic controls for heart rate? All right, let's get into it. So intrinsic control. When it comes to intrinsic control of heart rate, it's the nodal tissue. In the heart, all the cells in the heart depolarize at a slow rate. And once they depolarize enough, they have a muscle action potential, which is when the muscle contracts. There's specific tissues within the heart called nodal tissue that depolarize faster than the rest. The fastest one sets the heart rate under normal circumstances, because when the fastest one contracts, that wave of depolarization spreads out through the through the heart, and causes all of the other ones to depolarize at that moment, resetting their timer. This is called overdrive suppression. So the SA node, the sinoatrial node, firing is genetically set, but it does provide the basic rate to the heart. So in this sense, it is a factor. However, there's little evidence that the heart can regulate its own rate through the SA node by making adjustments. Thus, it seems that all adjustments come from extrinsic mechanisms. So there is a base rate, a base depolarization rate of the SA node. The heart does not need signals from the other parts of your body to note a beat. It's got this base rate via the SA node. Now, signals come from other parts of our body. Those are the extrinsic controls that can affect this base rate, can speed it up or slow it down. Let's look at the extrinsic controls. So we've got the autonomic nervous system. Both parasympathetic and sympathetic fibers go to the sinoatrial node, the SA node, and the AV node. These are independent branches so that they are controlled separately. Because of tonic control, what's tonic control? There's always some level of stimulation. So under normal circumstances, there's always some level of stimulation. There's always some frequency of parasympathetic signals going to the SA node, and there's always some frequency of sympathetic signals going to the SA node. When the parasympathetic frequency increases, heart rate decreases. It's inversely proportional. When sympathetic stimulation increases, heart rate increases. It's directly proportional. Hormones. All of those studied have positive chronotropic effects. So all hormones that we know about, that we study, increase heart rate. They have positive chronotropic effects. A positive chronotropic effect means you increase frequency, increase the heart rate. Specific examples are epinephrine and norepinephrine, the sympathomimetics because they're from the sympathetic nervous system, they're neurohormones, epinephrine and norepinephrine, which stimulate beta-1 receptors in the heart that have a similar effect of that to sympathetic stimulation. Thyroid hormone, T3 and T4, increase body temperature and heart rate. They do a lot of things. Thyroid hormones do a lot of things. Other extrinsic controls, heat has an influence on heart rate as well. Heat increases body temperature and leads to an increase in heart rate. This is often due to an increased demand for oxygen. All right, let's move on to number 2. The inotropic effects of the heart in many ways determine the stroke volume, which is a very important facet of blood pressure control. However, there are other factors that exert control over stroke volume in the heart. Specifically discuss the important intrinsic and extrinsic factors that alter stroke volume. So this is a similar question, intrinsic and extrinsic controls, but it's for a different aspect of heart function. It's for the inotropic effect, the strength of contraction. That's going to affect stroke volume. So let's see. Intrinsic controls. So the Frank-Starling law of the heart. You should have a flashcard for this. It should be underlined and bolded. It's the intrinsic autoregulation. It's based on the volume of blood in the ventricle before it is ejected. The greater the volume, the greater the stretch of the heart. This leads to an increased number of cross-bridging. Remember, cross-bridging is what generates force within the muscle cell, and a stronger contraction, which results in increased blood ejection. Stronger contraction, more blood is pushed out of the heart. The stroke volume is increased. If you increase stretch, you increase the sarcomere length, you increase cross-bridging, you increase contraction strength, you increase stroke volume. Remember that it is the ESV. What does ESV stand for? End systolic volume. That is highly regulated, and it is regulated by this concept, Frank-Starling law of the heart. Preload is a little kind of similar, actually. Preload is the pressure of the ventricle before ejection. If the pressure of the ventricle is higher, there's a greater pressure difference for ejection. So there is greater ejection from the ventricle. Remember, it's pressure differences that dictate flow, the amount of flow that you have. This is independent of Frank-Starling, but the most common mechanism that increases pressure is an increase in blood volume. So an increase in blood volume is the most common mechanism that increases pressure. Extrinsic controls. We're going to have the autonomic nervous system again. Now, this is primarily sympathetic, as there are few, some say none, a few parasympathetic fibers that innervate the ventricles. Increasing the sympathetic activity increases inotropy by allowing a greater calcium conductance. Greater calcium conductance leads to more cross-bridging and greater contraction strength of the heart. So there is an increased stroke volume. Remember, remember back to the frog lab. the more calcium that we get released into the cytoplasm of the muscle cell, the more cross-bridging, the more force that can be generated within that muscle cell. Hormones, epinephrine and norepinephrine, stimulate beta-1 receptors. This will increase stroke volume. Afterload, so the amount of pressure pushing back on the ventricle from the aorta or the pulmonary artery. Remember, no blood leaves the ventricle. No blood leaves the left ventricle and moves into the aorta until the pressure in the left ventricle is higher than the pressure in the aorta. If that aortic pressure is higher, let's compare aortic pressure of 80 to aortic pressure of 100. As we're going into isovolumetric systole, isovolumetric systole will end at 80 pressure when the pressure in the aorta is at 80. Isovolumetric systole will end at 100 when the pressure in the aorta is 100. It has to overcome the pressure in the aorta in order to push blood out of the left ventricle. This is afterload. If the pressure increases, there is a greater pressure the ventricle must overcome in order to eject blood. Thus, it is inversely related to stroke volume. As afterload increases, stroke volume decreases. Venous return. If you don't have any blood in your pump, you can't pump blood. In order to have cardiac output, venous return needs to continue as well. This is accomplished through venoconstriction, the muscle pump and the respiratory pump. What is the muscle pump? Remember this one? So this is when we contract our muscles, it squeezes and puts pressure on the venous system, which increases pressure locally in that vein and will help push blood back towards our heart. Calcium conductance. As we increase the entry of calcium into the cell, we will have greater stroke volume due to increased cross-bridging. Perfect. All right. Over to number 3. Explain both intrinsic and extrinsic mechanisms that control TPR or total peripheral resistance. First up, let's talk about the factors of resistance. Be familiar with these. Know this next part. So, what affects resistance in our circulatory system? Well, we've got Paisuli's law, which quantifies resistance in a tube. I don't speak French. I'm probably saying that wrong. This person's law quantifies resistance in a tube. So here's the equation. R is equal to 8L times N divided by R to the 4th. Radius is to the 4th. So a small change in radius can have a huge effect on resistance. All right, so tube length. Resistance is directly proportional. Longer tube, more resistance. Can we change our tube lengths? Yes, we can. You put on 20 pounds, you're going to have more tube length within your system. Can you quickly change tube length to change your blood pressure from moment to moment? No. Viscosity of fluid. Resistance is directly proportional. So thicker fluid increases resistance. Does viscosity of our blood change from time to time? Yeah, if you get really dehydrated, it might go up. If you drink a bunch of water, it might go down. Is that a good way to control our blood pressure, though? Can you quickly change the viscosity of your blood? No, it's not a good way to control our blood pressure. Tube radius. Tube radius resistance is inversely proportional, so a larger radius decreases resistance. Can we quickly control tube radius? Yes, we can via our sympathetic nervous system. And it has a huge effect on resistance because it's raised to the 4th power. So variable total peripheral resistance, or TPR. Intrinsic, the myogenic mechanism. So the arteriole controls its own diameter by responding to stretch or pressure exerted on the walls of the vessel. So the muscle cells within the cells within your arteries will feel stretch come and will resist that change. Myogenic mechanisms. It'll change the amount of force they're generating based on how much pressure is coming through the artery in order to help maintain level blood pressures. Chemical mechanism. Arterial diameter is changed by chemicals such as adenosine, carbon dioxide, histamine, lactic acid, potassium, nitrous oxide. All of these local chemicals, these are going to be your paracrines and autocrines and chemicals that are produced locally, not hormones which are produced in a far off area locally. They're vasodilators. A lot of them are actually byproducts of metabolism. Extrinsic control. So autonomic nervous system, but sympathetic only. Sympathetic only. Sympathetic only for our blood vessels. Hormones. Epinephrine stimulates alpha-1 receptors in vessels that have a similar effect to that of sympathetic stimulation. Vasoconstriction. All right, everybody. That's it for group problems 1, 2, and 3. I will see you in part 2 for the second half of them. As always, let me know if you have any questions, and I will see you in the next video.